Hi everyone, we hope you are enjoying the 2021 SETAC Europe meeting. For our featured presentation, we'd like to share with you an exciting new literature piece we've developed to showcase our expansive offering of standards to support biomonitoring studies. To begin with some background, CIL is a leading manufacturer of stable isotope labeled standards for environmental, food, water, and exposure analysis, commonly referred to as Exposomics. CIL recognizes that many organic contaminants are not only environmental pollutants, but may also be food and water contaminants and ultimately compounds of concern in human exposure studies. Our goal is to develop standards that have been identified as contaminants that may pose a risk to human health. CIL has and continues to produce standards which fit the needs of specific testing programs such as the Center for Disease Control's National Health and Nutrition Examination Survey, commonly referred to as NHANES, the National Institute for Environmental Studies Japan Environment and Children's Study, commonly referred to as JECS, the European Human Biomonitoring Initiative, commonly referred to as HBM4EU. This is comprised of 30 countries, the European Environment Agency, and the European Commission, and the Institute for Exposomic Research at the Icahn School of Medicine at Mount Sinai. The emergence of new research institutions and programs is ongoing and a testament to the importance of studying the impact the environment can have on human health. Investigating the different routes of human exposure is essential to biomonitoring research. Here we have outlined the main sources of contamination and their common exposure routes. Dermal exposure is widely experienced with application and use of personal care products and pharmaceuticals. Ingestion exposure is commonly experienced with food and food packaging contaminants, drinking water, pharmaceuticals, tobacco products, and personal care products. Inhalation exposure is commonly seen with industrial pollution and tobacco products. Now that we've covered the routes of exposure, let's talk about specific product classes of focus with biomonitoring studies. The classes we will discuss include compounds produced via industrial processes that have been detected in consumer products, food and food packaging, water, pharmaceuticals, and tobacco products. CIL offers stable isotope labeled and native standards supporting each of the product classes we will mention, and in some cases also offers multi-component mixtures. Our first product class of mention is biomarkers. While sources of exposure are expansive, including air, food, water, drugs, etc., examples of biomarker classes of environmental contaminants include PFAS, tobacco-related compounds, PACs, herbicides, insecticides, and pesticides, dioxins and furans, PCBs, flame retardants, and steroids and hormones. Diving into some of these biomarker classes a bit further, let's start with PFAS. CIL offers stable isotope labeled and native standards for many of the traditional PFAS classes, as well as emerging fluoropolymer classes. CIL also offers several native and labeled multi-component standard mixtures, including US EPA Method 537 and 537.1 mixes. For our tobacco-related compounds, we offer tobacco-specific nitrosamines, nicotine, tobacco additives, tobacco biomarkers, and emerging biomarkers in e-cigarette use. For phthalates, CIL offers diester and monoester phthalates, primary and secondary metabolites, terephthalate esters and metabolites, DINCH, MINCH, trimalidic acid esters and metabolites, and a multi-component mixture. Within the PAC category, CIL offers PAHs, PCNs, alkyl PAHs, halogenated PAHs, hydroxy PAHs, nitro PAHs, amino PAHs, as well as several mixtures. For phenolic compounds, we offer halogenated benzene, phenol, and anisole standards, alkylated phenols, nitrophenols, and mixtures combined with parabens. CIL offers a variety of bisphenol standards as well as related compounds including glucuronide conjugated, bisulfate conjugated, and bisphenol diglycetyl ethers. For parabens, we offer paraben and paraben metabolite standards as well as mixtures combined with phenolic compounds. We continue to add to our already extensive inventory of pesticide standards and are focused on producing metabolite standards as studies focus on exposure effects and metabolic pathways of parent compounds. Our expansive listing includes Stockholm Convention compounds including organochlorine pesticides and toxaphene, organophosphate and carbamate insecticides, crop protection standards including glyphosate, glufosinate, AMPA, dicamba, and polyethoxylated taloamines, neonicotinoids, pyrethroids, triazine herbicides and metabolites, fungicides, pesticide standard mixtures, cannabis pesticide standards, and pesticide and herbicide metabolites. I'd like to note that CIL's origins date back to making some of the first dioxin standards for testing purposes. We offer polychlorinated dioxins and furans, method standards to support US EPA methods 1613, 23, 8290, and 8280, European Air Method EN 1948, JIS Method K0311 and K0312, mixtures containing dioxins, furans, and PCBs, food and feed mixtures, brominated dioxin and furan standards and multi-component mixtures, and native qualitative solutions for elution profiling. CIL offers PCB standards and standard mixes, method standards to support US EPA Method 1668C, CEN Method EN 1948-4, WHO dioxin-like and non-dioxin-like mixes, mixed bromo-chloro standards and standard mixtures, and combined dioxin, furan, and PCB mixes. For flame retardants, we offer BDE standards and standard mixes, BDE metabolites, US EPA Method 1614 standards, ROHS BDE standards, alternative halogenated flame retardants including tetrabromobisphenol A and tetrachlorobisphenol A, declaranes, Firemaster 550 standards, and hexabromocyclododecane standards, as well as polybrominated biphenyls and phosphate flame retardant standards. We've recently overhauled our former Exposomics flyer and are very pleased to present to you our brand new biomonitoring brochure. We have included all the product classes mentioned today as seen in the page excerpts throughout this presentation. We've also highlighted some of the specific targets of Exposomics researchers within each class. The brochure can be viewed and downloaded in our virtual booth. Once in the PDF, each section has links to other relevant literature pieces as well as the corresponding product class page of our website. We hope you find this comprehensive piece useful for your research applications. Thank you for your time today, and as always, please feel free to reach out if you have any questions or to discuss your standards needs with us.